Thank you, Xinyan, to organize this symposium. And there's still a huge gap between population growth and food supply, and a lot of developing country, facing a serious food insecurity. But the availability of arable land reached a plateau, and the increase of food supply will rely more on technology, in particular plant breeding. You must think of China as second largest economy has secured food supply. But in fact, we import almost 100 million tons of soybean, which would occupy more than 760 million mu or 50 million hectares of arable land if grown in China. And we transport 100 million tons of grain from North China to South China, which equals the use of 100 billion cubic meters of water, more than 2 times of south to north water diversion project. This is a mega project in China to ship water from south to north. You know that North China, the water is insufficient. Let's look at the crop we grow. In fact, there are 2 types of crop. In the top 100 crop with largest acreage, 55 are sexually propagated by seeds, such as rice, maize, and wheat. And these crops are fast-breeding crops. And the other 45 are asexually propagated, clonal propagated, such as potato, cassava, and sweet potato. These are slow-breeding crops. Many of these slow-breeding crops are the staple food of developing countries. For example, cassava is the major staple food in Africa. The genetic potential of these slow-breeding crops are largely untapped. Potato is the number 3 staple food worldwide, feeding over 1 billion people in over 100 countries. It's quite nutritious and healthy food, yet it's clonally propagated with highly heterozygous genome. That means the genetics analysis and breeding is very slow. Here I wish to draw a comparison between car business and potato breeding. 120 years ago, Mercedes looked like this. Now it's very fancy. And 120 years ago, Receivable Bank is already a major variety in the United States. It's still the number 1 variety in the United States. In addition, the propagation index of potato is lower. About 10% of the total yield needs to be kept as seed potato for next year. The price for seed tuber is high. It takes $1,000 to $2,000 per hectare. Besides, the tubers often carry some disease and pathogens and pests. And our questions are, can we accelerate the breeding efficiency by changing the reproduction strategy of a crop? And can we make potato a more breeder-friendly and farmer-affordable crop? A few years ago, we initiated the U-Potato Plan, which is to transfer potato from a clonally propagated tetraploid crop. Tetraploid means to have 4 sets of chromosomes, into a seed diploid crop, reducing the breeding cycles from 10 to 12 years to 3 to 5 years, and increase the propagation coefficient 1,000 times. Normally for 1 mu is a unit of acreage in China. That's 50 mu is 1 hectare. So for 1 mu, we need 200 kilo tubers as seeds. And now we only need 2 grams of seeds. And I happen to carry 2 grams of seeds in a tube. That's the need, the volume of seeds needed for 1 mu. 15 of this will grow 1 hectare. We will utilize heterosis, hybrid figure, in potato. Heterosis was I think first recorded by our agronomist Jia Sixie many years ago. In 齐民要素 is China's first agricultural encyclopedia. Moors shows higher figure than the parents' donkey and the horse. And Charles Darwin also described the figure of hybrids by many experiments. Of course, we can learn from hybrid maize breeding. Between 1910 to 1970, the yield of maize increased by 8 times through hybrid breeding. Without hybrid maize, we would have no modern agriculture and no modern civilization. Yuan Longping and his team successfully increased rice yield by 50% using hybrid breeding. He's my hero. What is science behind the rice heterosis? Han Bin and his team collected thousands of hybrid rice varieties and analyzed their genome. And they discovered that 16 core genetic genes contribute over 70% of heterosis. And these findings are of great significance in promoting design breeding of hybrid rice. For hybrid potato, the challenges are even bigger. It involves the transformation from tetraploid to diploid, meaning packaging genes from 4 sets of chromosomes into 2 sets of chromosomes, from asexual to sexual reproduction, from tuber to seeds. And its genome carries a lot of deleterious mutations and structural variations. Hybrid potato requires a deep understanding of the potato genome and the strategy of genome design. Here I would like to talk about our progress in 3 parts. First, we developed a deep understanding of genome variation and evolution of potato. We decoded the hybrid genome in 2011. That's 1X. And we recently sequenced the 2X and the 4X genome of potato. Hybrid potato requires a broad genetic diversity, and we therefore developed a high-quality potato pangenome. Pan means a collection of genomes, diversity. Applying multi-omics and evolutionary analysis, we discovered the organ identity gene of tuber, IT1. If this gene is damaged, it won't have any tuber. And tuber is a new organ, newly evolved organ. It doesn't exist in the cyst clade tomato and E. tuberosum. Potato and tomato are cousins in this sense. And we discovered IT1 received a signal from the leaf sp6a, move downward to the underground on the stem to make a tuber. Remember these 2 elements. I will talk about that later. And we discovered quite a mess in the evolution of potato. There's frequently interspecific crosses and gene flow. And this led us find a very interesting, will have a very interesting discovery that the potato it itself is a hybrid of tomato and the E tuberosum. And the mitochondria genome told us tomato is the father, is the mother. E tuberosum is the father. And the hybridization occurred around 9 million years ago, coincide with the rapid uplift of the Andes mountain. And the hybridization brought SP6A from tomato and IT1 from E tuberosum and create the tuber, which can survive in the high mountains, because in the high mountain, the temperature can be low, not good for flower. And the tuber is then a very useful alternative for survive the plants. And these events give rise to a major staple food crop. How lucky we are. And the hybridization events temporarily, we think, destroyed the stability of genome, and thus lead to the birth of transposable element, TE. And we found the potato genome, in the potato genome, 90% of the structural variation are associated with TE. Because they have very active TE, it have a lot of structural variation, make the genome very, very, very dynamic and polymorphism. About 1.5 of the genome are found to be heterozygous SNPs, single nucleotide polymorphism, which is much higher than rice. Rice is about 0.5. And also surprisingly, about 13% of the potato genome are defined by structural variation. So added together, 15% of the genome are different between any two lines, between any two potato variety. And the inversion is one major type of structural variation and inhibit recombination. We therefore create an inversion map to avoid linkage drag. Linkage drag is a kind of not so good for breeding. When you want to use one gene, you always carry a lot of genes around that gene. So to make a hybrid, we need to develop two highly homozygous inbreeding lines as parents. And the major obstacle in inbreeding line development is deleterious mutation. Clonal propagation facilitates accumulation of large number of deleterious mutation, which can affect any traits. And this deleterious mutation often hide in heterozygous status. And because it's clonally propagated, it doesn't have any chance to become homozygous. You need to surfing to become homozygous. Normally, larger effects, deleterious mutation can be purged during surfing. However, when two deleterious mutations are tightly linked in repulsion phase, it is very difficult to eradicate them. The so-called Hill-Robertson interference, unless a crossing over between them was discovered. And that explains why some line cannot reach a high homozygosity. For example, this line has been self-crossed for 9 times. It still have 20% of the genome, more than 20% of the genome becoming heterozygous. Normally, when you self-cross a line for 6 times, it should reach the homozygosity of 99%. Even the fully homozygous inbreeding line, that's F1 and two inbreeding line besides it. Even they don't have larger effect deleterious mutation, it still carry a lot of mild effect deleterious mutation, which jointly resulting negative fitness results, as can shown by frail growth, reduced fertility, and low yield. So it's imperative to identify all deleterious mutations and quantify their effects on fitness. However, the current algorithm, SIFT, can only predict non-synonymous deleterious mutation and cannot quantify their effects. We therefore apply a new method, genome evolutionary rate profiling GERP, to quantify constraint size. But first, we need a deep phylogenetic tree. We develop a deep phylogenetic tree of 100 species in Solanaceae. Solanaceae is the family, botanical family of potato, tomato, so far. And we made alignment, whole gene alignment. And this phylogenetic tree covers 9 of the 13 clades of Solanaceae. And the total branch length is 4.05. That represents the number of substitution in ancestral allele at the neutral site. And this deep phylogeny ensured identification of evolutionary constraint sequence in the botanic family. It looks like a lens, evolutionary lens, to see in the potato genome during the 80 million years of evolution, which bases are very conserved, they don't change, which bases are quite evolved. So it's a differentiate, this lens differentiate the most constrained, most conserved from most changed. Using GERP score of 2.75, we discovered 2.4% of the potato genome are constrained. A mutation on these constraint sites tend to be deleterious in potato. Out of this, 36% constraint sites are in non-coding region. And this data set combined with the variation map of 192 potato germplasm, we create a two-dimension map for potato. On this side is a genotype dimension. On the other side, vertical side, is the deleterious dimension. So on each base, you know which base is polymorphic, and you also know whether this base is deleterious or not. We found that 50% of deleterious mutation in non-coding region, and in the coding region, 1/3, totally 15%, are synonymous. We identified a deleterious allele, small FBA, which is control flower abortion, which contains a causal deleterious mutation in its promoter region. They cannot identify using the previous algorithm. So prediction of deleterious mutation can help us on hybrid potato breeding. And we sequenced these 192 potato lines and adopt a formula to calculate the whole genome deleterious burden, that the total amount of deleterious mutation and times their effect. This leads to a counterintuitive proposal. Normally we select the good-looking lines for breeding. Now we found the weaker lines carries less deleterious burden, which are in fact a better choice for developing inbreeding lines. So it's counterintuitive. And we incorporated this deleterious mutation map into genome prediction and increase the prediction accuracy for up to 45%. And when our paper released this year, and at the same week, there's a series of papers published in Science on the analysis of evolutionary constraints in the mammal genome, and also the retention mutation for human disease study is quite similar to our study. With all this knowledge and tools, which we perform the genome design of hybrid potato breeding. But we still have to overcome one of the obstacles to break the self-incompatibility of potato. And this self-incompatibility is a two components system, sRNS in the stigma and F-box in the pollen, resembling the lock and the key. Weirdly, its own key cannot open the lock. So the strategy is that we either break the lock or we find a master key. We break the lock by genome editing of sRNS and also find its natural mutants. On the other way, we found the gene of master key, the SLI gene, encoding a new F-box gene, F-box protein. And this discoveries led us to create self-compatible potato. With all these tools and information ready, we developed the first generation of a highly homozygous inbreeding line, up to 99 to 100%, using genome design, which involve 4 steps. First, selection of starting material of a high genome homozygosity. And second, genetic analysis of the segregation population for purging of deleterious mutations. And 3, breaking tight linkage of deleterious mutation to develop an elite line. And 4, crossing inbreeding lines with high genome complementarity to generate vigorous hybrids. And these 3 plants on the left are the mother plants, and those 3 are the father plants. They are not from tuber; they are from seeds. Looks quite similar. And then in the middle are the F1 hybrids, showing very strong heterosis. And this is our prototype hybrid U potato 1. It has already over 50% of the yield of commercial variety, showing a strong heterosis. Increased, they also have rather high dry matter content and high carotenoid reaches content. Tastes very good. Of course, they also have some disadvantages. Remain in this first generation is prototype. Requires continued genetic improvement. And we are developing a fast iterative breeding program for U potato series. So this is the second generation will have new traits of a good tuber shape and a long dormancy period. And for U potato 3, we have late applied resistance and long day condition, adapt to long day conditions. And for U potato 4, we will have cytoplasmic male sterility. The potato doesn't need to have flower for the F1s. They can save the energy to make a bigger tuber. And for U potato 5, we have high yield and stress tolerance. And each cycle will take about 2 or 3 years. So they really move to this fast track of potato breeding. This is a U potato 1, where it looks like this. And the U potato 4 is the adding of 8 genome segments, and plus a cytoplasmic male sterility. These 9 components will make a U potato 4. We have a time plan for 20 years in 4 sections, and we are now in the second 5 year. Eventually, in this 20 years, we will make a full commercialization of this hybrid potato. And my team is now visiting Rwanda, planning small scale of field experiment there. And we also trained one of the PhD students there, and hope they will become a Rwanda potato breeder for hybrids. And our research received quite some positive comments. Professor Pete Morrow from University of Minnesota, he's a maize geneticist. He mentioned that it was one of the most ambitious and perhaps audacious projects. But it's quite a pleasure to discuss hybrid potato with Yuan Longping. He's the father of hybrid rice. That's half years before he's passing away, and I hope I can show.
种子繁殖技术还可以改进那个马铃薯的这个病毒的问题。原来湖南省种马铃薯在内蒙去搞种子，麻烦。马铃薯种子繁殖是革命性的技术。
I mentioned that hybrid seeds can solve the problem of virus, and also South China don't need to transport potato from North China as seed potato. The plan is a multi-group effort. I wish to thank all my colleagues, in particular Chen Zizhang from AGIS, Shang Yi from Yunnan, and Dong Songmeng from Nanjing. International Center of Potato contributed many germplasm. And I thank the Ministry of Agriculture, Ministry of Science and Technology, and SFC, Guangdong and Shenzhen for funds. Thank you.